I just want to start by asking for you to tell us a little bit about yourself.
So I first got interested in biological sciences as an undergrad because of an interest in ecology and conservation biology. And that led me to my first experience in microbial ecology, where as a master's degree student, I was using a combination of molecular and cultural techniques to study a type of fungi in soil, Chytridia mycota, the order Chytrids. I remember at the time my advisor said, no one's studying these things. It's wide open. And I questioned the importance. Well, why isn't anyone studying this? Surely they must not be important. And meanwhile, at that time, there were these frog die-offs going on around the world that were unexplained. And it turned out that it was a type of the fungi that I was studying, chytrids, that were causing it. And that was a real eye-opener for me that, just because it's wide open and no one's studying it in the microbiome world doesn't mean it's not important. In fact, there's so much we don't know about the microbial world and its importance in all sorts of different systems. And so that was my first spark of interest in microbial ecology. But then after that, I actually went to a molecular evolution lab as a research assistant at Princeton for a bit. And that's where I was first introduced to the world of bioinformatics and molecular evolution and got a really great interest in that. So when I decided to go for my PhD with Rob Knight, I was really drawn to this project that combined microbial ecology with molecular evolution and bioinformatics. And we made a tool for analyzing microbial communities using their information in their phylogenetic lineages called UniFrac. And UniFrac became kind of a standard in the field, which is extremely exciting to me to see so many people using the tools we built fruitfully. But another thing I really always enjoyed doing was taking data in the public domain and using UniFrac to tie them together to generate new insights about what were the main environmental and biological factors that seem to differentiate microbes that lived in different environments. And so I really have a great interest in how we can take data in the public domain and learn new things from it is another interest of mine. Another thing that starting in my PhD work I became very interested in is the human microbiome. And that was really from interacting with Jeff Gordon at first. Actually, the very first application of UniFrac in a paper was the very first paper out of the Gordon lab that showed a correlation between the gut microbiome and obesity in a mouse model. And I really got interested then in the human microbiome and its roles in health. And ecology, just always viewing it still as there's this ecosystem of life living in your gut, but now really interested in how that interacts with the host. And so now I have my own lab and really draw a lot from my experiences both working with Jeff Gordon and Rob Knight. So kind of as coming out of the Knight lab, we're really still doing a lot of informatics, a lot of technique development for analyzing data, but really try to integrate that with all sorts of experiments in notobiotic mice, with cultured isolates in vitro, to try to get at mechanism. And I'm a real big proponent of team science and multidisciplinary research that can help us get a really deep understanding of the full microbiome system and how it interacts with the host.
Are there any areas of science that particularly interest you just now?
Yes, absolutely. The microbiome field has been so fun to watch evolve through the years. And I feel like with next generation sequencing, there was this, I would say we're still in the middle of a massive trajectory of initial understanding and characterization just of what's there, what are the functions. And one of the exciting things about the field to me now is the new tools and the ability we have, given a lot of this baseline look at the composition of the microbiome, to move more into mechanism, to move into how mechanistically do microbes interact with each other in a community, how do they shape ecosystems, how do they shape host health. And so one of the things I mentioned earlier is I'm a big fan of seam science. So stories that really integrate a lot of different omics with experimental validation and a variety of different expertise of different PIs is very exciting to me. I feel like a lot of papers are really getting to the point that go beyond, we recognize an association to really understanding the mechanisms underpinning how microbes are interacting with each other, with the environment, with the host, in driving that relationship. Another thing, I will say, I think that working in human biology and working in environmental systems in some ways have different challenges and interests. So, human microbiome at this point, I feel like there's been advancements not only in omics, but in being able to culture more of the different organisms that live there, building huge databases of reference genomes that we can use. Whereas environmental systems, in a way, are way more complicated in that there's a greater diversity of them, and many of them are individually just more diverse, and less of the microbes have been cultured. And I think there's a lot of room for innovation there in the omics technologies, the shotgun metagenomes. I mean, a paper that might excite me is one, well, this whole new lineage of bacteria we didn't know about performs this essential function. And those sorts of very basic, what does the tree of life do, discoveries are still coming out of natural systems. And I think that's really super exciting as well. And a third aspect really that excites me is just papers that are well grounded in theory, like ecological theory, and using a strong theoretical background that's really grounded in the depth of literature in that field to explain ecosystems and their function and impact.
What was it that made you want to be the editor-in-chief of MPJ Biofilms and Microbiomes?
So I was very excited when I got the invitation for this opportunity. For one, I feel like the scope of the journal is so well within my background and interests on a couple different levels. For one, just having delved in both the environmental world and the human health side of microbial systems, I love it that NPJ Biofilms and Microbiomes, we can get submissions from people in both of these different backgrounds of interest. I'm really interested in how microbes interact with each other in biofilm communities, but also how they interact with other hosts, human and otherwise. And so really the scope of the journal in terms of it's looking at these interactions between microbes and other organisms, and being able to be in a situation to push forward that sort of research is very exciting to me. I also, throughout my career, as I mentioned, has really loved looking at microbes in all systems as ecological communities and looking at ecological theory as a backdrop to understand these complex systems. And all of that seemed very within the scope of this journal. I'm excited that I love to read and I love to write, and I just feel like an editorial sort of position is a great fit for me. And I also love going to meetings and learning about new and exciting science. And it's exciting to me to have the opportunity to be at a meeting and be like, man, that was a great talk. That's such exciting research. And to be able to reach out to whoever gave that talk and be like, hey, would you like to write an article for our journal? And just to have that platform to give people doing exciting research the opportunity to share it with other people.
And so then, what are your aims going forward with the journal?
Well, it's an exciting time for the journal. So I'm really excited to work together with my co-editor-in-chief to push forward both the microbiome and the biofilm aspects of the journal. So Elaine's going to be focusing on the biofilm submissions and myself on the microbiome submissions. So my aims are really to make this a world-class journal where individuals who come to the journal feel that they can get access without exception to high quality work where they're not looking at who reviewed this paper and and for also the authors who interact with the journal to have a good experience. Overall, I hope that the journal becomes an outlet for research and exciting new discoveries in the biofilm and microbiome arena, and that readers and authors have a great experience publishing with us.